Hi, Tom Roseberry, CTO and co-founder at Oleo Labs. My background is mechanical engineering, then going to triathlon coaching, then moving to neuroscience, wondering why racing was really fun, training was really boring. From that, went to Neuralink, was one of the early employees there, and through a series of startups, worked my way towards therapeutics, most recently at Loyal as the vice president of engineering, computational biology, and drug development. My co-founder David and I started Oleo Labs about 2 years ago. Trying to work on how you can actually build and predict combination therapeutics that are going to be better, have better efficacy with fewer side effects. And the idea here is basically that the body is really complex. This has been a theme actually throughout the entire day here. There are thousands of different interacting proteins and cell types throughout the body, and a single therapeutic or a single target doesn't really approach most of the diseases that we experience. And so this is actually already happening in the real world, that about 40%, can't really see that, but 40% of Americans are already taking multiple therapeutics at the same time. So can we actually predict and better kind of de novo build combination therapeutics for the individual? So we built a platform around this. There are 3 different components of it. We wanted to know how people were experiencing their therapeutics or their diseases in the real world. So we got access to a 120 million patient data set, EMR type data. We then built whole body AI models that actually can predict the effect of a combination of drugs on the human body given a patient population. That whittles down the trillions of different combinations that we could actually try. And then we built a custom rodent platform where we can actually observe the behavior of mice, and that maps actually back to human data quite well. This is a repeatable process for many different indications. We actually in-license molecules or repurpose rather than build molecules. We let you guys all do that. And this actually reduces our time from clinic from 5 to 2 years. So basically how these kind of fit together is that we mine that EMR data set for how people are experiencing either disease or their therapeutic. For example, a great example of this is actually we just published a bioRxiv paper on how the mechanism for why women experience 2.5 times the side effect rates on a GLP-1 drug than men. We actually went down to the single-cell RNA sequencing for that and established a really nice mechanistic model for that. We then build that model in silico so we can test combinations in silico, and then we test those combinations, the best combinations, in our rodent platform. That rodent platform is explicitly transformed back onto human data to make sure that what we see in the rodents is actually what will likely happen in the real world. So just to outline the human EMR data, we started in obesity, and there are 2.2 million people that have taken a GLP-1 drug in this data set. We use it to validate our rodent and in silico models and actually, again, identify underserved populations. Our in silico whole body models are actually kind of like a filter, where on one side we can perturb proteins, and on the other side we get the effect of that protein perturbation on a phenotype or a behavior. For example, hunger, thirst, glucose levels, things like that. In the middle are a number of layers that kind of, as I was saying, act like a filter. Some of them are straight biological data that have been normalized to be able to function with the other layers. And then the other layers are kind of an AI-based compression of literature where we've, as experts in neuroscience, basically the entire company are a bunch of neuroscientists, we've created data sets of our expert assessment of neuroscience literature, trained an LLM on that, and then extrapolated that out to the rest of the proteins and cells in the body to create kind of an atlas of what happens when I either activate or deactivate this protein. We then built this rodent platform that's actually trying to take a cell culture approach to rodent work. We built 80 boxes. We should have built 96 for the 96 well plate, but we missed. These boxes have a video camera on top, a thermal camera, CO2 sensors, ammonia sensor. The mice are recorded 24/7. We've actually been able to do an experiment in 24 hours that maps onto what happens to a human at 40 weeks. We've now screened through 250 different drugs, and we're actually able to predict clinical trial success. This is out of date. We're at 0.89 ROC on clinical trial failure prediction. Yeah, and so one of the big things that rodents have been very not good at is predicting nausea or being able to identify nausea in a rodent. Oftentimes, people try to use pica behavior, which is eating non-nutritive substances like kaolin clay. We just did an explicit transformation from our rodent behavior, taking each rodent produces 2.5 million data points throughout the day. We took those data points and just mapped it directly onto human data and can get a very nice nausea prediction at various doses of the current obesity drugs. So how do these platforms all fit together? How well do they perform? If we take current obesity drugs, liraglutide in green, which is one of the early Trulicity drugs, semaglutide is Ozempic, CagriSema is a phase 3 Novo Nordisk combination. Tirzepatide is Mounjaro, which is Eli Lilly's molecule that's actually taking over Ozempic. Retatrutide is their triple G or triple agonist that's supposed to take over tirzepatide. We get a strong correlation between the compute, the rodent, and actual human outcomes. That rodent data, again, is 24-hour recordings that are mapped onto 40-week human data. So we have 3 different pipelines running right now. One, OL1 is to mitigate muscle wasting when taking a GLP-1. We actually think that could be an amazing aging drug for sarcopenia. OL2 is trying to mitigate nausea when taking a GLP-1 by reducing the amount of GLP-1 you have to take. And then OL3 kind of goes back to that bioarchive paper that we just published, trying to build a combination specifically for women to mitigate the side effects. So we're funded by some amazing investors and have some really great advisors on board, and the team has been doing amazing. Yeah, that's pretty much it. Go check out that bioarchive paper.
We have a question. Great talk. From your EMR data that can be super messy, I'm curious what you see of drugs that patients may not be reporting that they're taking, supplements they're not reporting that they're taking. How are you dealing with the messiness in the EMR data, or people who are taking off-label or compound pharmacy of the Ozempic drugs?
Yeah,
or GLP-1s?
That was a really tough. We see a lot of discontinuation, but the discontinuation on GLP-1s could be from somebody actually discontinuing or going to a compounding pharmacy. We don't capture that, and so what we would do with a data point like that is we'd set a discontinuation day and just not look at the data after that. If we were measuring something around GLP-1s, for example. So you have to be incredibly careful with the EMR data. We thought we had a really great combination that we had kind of mined out of it. Of a GLP-1 plus another drug that looked like it really drove a lot of weight loss. But when we did a lot more research on it, it turned out that doctors give this drug when somebody is experiencing rapid weight loss to mitigate like gallstones. So you have to be very careful with your conclusions.
Thank you. Next question.
Yeah, thank you so much. This is really exciting. I was wondering, I mean, in general, there's a huge problem in the industry that things fail in late-stage clinical trials. And it occurs to me that there's a large number of pharma companies that would be dying to have an R-squared of 0.08 on predicting that. And I was wondering if you've thought of, it sounds like you have your vertically integrated pipeline, but I was wondering if you've thought of, as another business model, expanding the space of what kinds of things you can predict clinical trial success on and addressing that problem industry-wide?
The answer is yes, and we are in talks with people around that. And we can actually, very early data shows that we can actually predict the type of failure as well.
Great. Thank you so much.